Well, first off, I'd like to thank the organizers for giving me this opportunity to tell you about how we're using modified AFM cantilevers to look at a variety of different biological systems. Shown here is a short RNA hairpin that's just 12 base pairs long, but we can easily see it open and close. Okay, just to give you an overview of my lab, we're using AFM initially to study membrane proteins, but we've sort of branched out to looking at globular proteins, protein ligand interactions, and more recently, nucleic acids, in particular, putting them down at biochemically relevant conditions. But membrane proteins has really been what drove us into studying or using AFM. Just so to highlight membrane proteins for those of you who are not familiar with them, they're really a frontier in structural biology. They represent 30% of the genome, about 1% of the PDB. They're fundamental to many processes of life, including transport, signaling. About 50% of current and future drugs target membrane proteins, and insights associated with solving structures of membrane proteins have been associated with 4 Nobel Prizes in chemistry. A typical membrane protein may have 7 transmembrane helices, shown here. So predicting the topology of how a membrane protein is inserted into the lipid bilayer is quite easy. What's much more challenging is understanding the energetics that cause it to fold. In part, this is because membrane proteins live at this interface where it's lipid or hydrophobic in the middle, it's hydrophilic on the outside, and there's a charged, highly charged interface in between. It's important to note that membrane proteins fold in equilibrated structures, even though they're inserted by the translocon. And so one of the fundamental problems is we lack precise measure of the energetics of membrane proteins in native lipid bilayers, or lipid bilayers in general. So many of you may be familiar with this assay. It was developed by Carlos Bustamante many years ago. Shown here is some data from our lab, as well as a cartoon illustration of an optical trap. And so when you pull on an RNA hairpin, you can easily measure it unfold and refold with great fidelity due to the precision of optical tweezers. And with this high signal to noise ratio, it's very easy to measure, let's say, the rate of unfolding and refolding as a function of force. Underlying these kinetics, of course, is the energy landscape. And as many of you know, we think of an applied force as tilting that energy landscape to the point where something that would normally be folded is now equally populated in an unfolded and refolded state. Now, one of the unique things about single molecule force spectroscopy, not just measuring the distance to the transition state or perhaps the height of the barrier, optical tweezers has been able to resolve a one-dimensional projection of the underlying energy landscape. So what we asked ourselves is, can we actually study membrane proteins with a similar level of control? And to do so, we've had to develop a series of modifications for cantilevers. So I will just briefly go over these. So our first insight was realizing that the force stability of AFM was degraded primarily by the gold coating on an AFM cantilever. So by stripping the gold and the underlying chrome off a biolever long, which is a very popular cantilever for force spectroscopy, we could achieve sub-piconewton stabilities over 100 seconds. Now, these long, soft cantilevers, they're 100 microns long, when they were in liquid near a surface, have a time response of approximately ½ millisecond. Our next step was to start using a focused ion beam to modify shorter cantilevers. These cantilevers are 40 microns long, and we left a small gold patch at the end for reflectivity, and we modified the shape to reduce the hydrodynamic drag. Now, these cantilevers now had a time response in liquid of just 25 microseconds. So 40 microns, we went down to 9 microns. So these are so-called modified biolever fasts. These are just 2 microns wide. Now, these cantilevers have exceptional time resolution in liquid, just 1 microsecond, but they suffered reduced force stability. And so for equilibrium assays, we're very interested in force stability. So we developed a sort of 4th generation cantilever, which my students called a Warhammer, which is based off of the same parent cantilever as shown here, but with a different head geometry. Now these cantilevers had 8 microsecond time resolution in liquid, but now have force stability of approximately a piconewton out to 100 seconds. So how do we look at that? Usually we look at the force precision as a function of averaging time, and this slope here is governed by Brownian motion, and you get below a piconewton of force stability at about a millisecond, and that goes out to typically around 100 seconds. To compare that to sort of traditional AFM reports of stability, it's usually about 5 to 20 piconewton of stability. So to give you a sense of what this looks like, what you see here is a scanning electron microscope of an ultra short cantilever that we're going to modify with a focused ion beam. Seen here, you can see that it's actually tilted, which is actually not good. We'd like the cantilever to be straight. And so what you see here is the process by which we modify it. First, we cut these slits, and then we're going to cut them a flap out, and it's going to go up, and then we thin it. And so the process of looking at this with a scanning electron microscope while modifying it was really important, because in that thinning step, you noticed that the cantilever straightened out, and that's very important to our yield. Okay, we want the cantilevers to be straight to be able to use them. Now, this process was developed by an undergraduate. Typically, we can produce about 2 to 4 per hour, depending on the size of the cantilever we're modifying. And I'd sort of like to emphasize that my lab had no prior experience doing focused ion beam modifications. So I don't think this is a technique that's too hard for other people to learn and adopt. So what we're interested in doing is applying this technology to the canonical membrane protein unfolding assay developed by Daniel Muller and Hermann Gaub many years ago. And in this assay, bacteriorhodopsin in its native lipid bilayer is mechanically extracted by AFM. And I'm sure many of you know the primary result from this work is that pairs of transmembrane helices unfold at one time. So we can zoom in on what some of that data might look like. So this is some later data from Daniel Muller's lab, again, force versus extension. In this case, we're pulling on the 3rd and 4th helices to get pulled out, the so-called ED helix pair shown down here. And what happens if you look in the red curve is that there's a force drop. This is associated with, and then another force drop. And the first force drop is associated with unfolding of the E helix, and the second force drop is associated with unfolding of the D helix. Now, it's a thermally activated process. Each time you do this, you go through a slightly different unfolding force. And sometimes you populate a different intermediate shown here. The take-home lesson here is that sort of state-of-the-art for the field for about 15 years was when unfolding a pair of transmembrane helices, you would see at most 2 intermediates. So now we're going to look at the same system with our modified cantilevers that provide 100 times higher time resolution and 10 times higher force precision. And so this is what you see. The first thing you notice, of course, is that it's much cleaner. This is a particularly simple unfolding curve. So if we do it again, we get a different unfolding pathway. If we do it again, we get a different unfolding pathway, and we can do it about 100 times in this paper. And each time it unfolds differently. So the take-home lesson here is that we went from seeing 2 intermediates to at least 14, or put differently, we were missing approximately 85% of the unfolding intermediates. Another thing that you can see is that in every single record that we look at, you can see that the force goes down and up and down and up, and this corresponds to local unfolding and refolding of the membrane protein. So this was unexpected because we were pulling at 300 nanometers per second. And so it's likely that the protein has always been unfolding and refolding, but just on time and distance scales that we haven't been able to see when using traditional cantilevers. So next we wanted to get to looking at energetics. and as alluded to by a previous speaker, one of the problems when you're trying to look at the initial unfolding of membrane proteins is that there's tip sample adhesion. And these types of assays, you take the AFM tip and you push it into the surface at about a nanonewton or two for 1 or 2 seconds, and then about 1% of the time you'll get a record that looks like this if you're lucky. But quantifying the initial unfolding is hindered by this tip sample adhesion. To address this problem, we engineered a cysteine into the tail of bacteria rhodopsin to allow us to do site-specific attachment. In this case, we converted that cysteine into a copper-free click chemistry reagent called DBCO, and we put an azide on the end of a short peg. And when we used this type of assay, now you can see a really clean initial unfolding curve. There's a new state that was not previously identified, and I'll talk to you about that in a minute. And there's some fuzz right here. And when you zoom in on that fuzz, what you see is beautiful 3-state near equilibrium dynamics. We can associate the top state with fully folded bacteria rhodopsin, and we can assign the next 2 intermediates. I'd like to highlight the time scale here is just 2 milliseconds. So these dynamics are happening on very fast time scales, and using worm-like chain analysis, we can actually deduce the distance between these different states. And this corresponds to the unfolding of just 2 helical turns of a single alpha helical helix. The bottom state here is associated with a retinol, and we can remove the retinol and change the stability of this bottom state, which verifies our assignment. Just to highlight the sensitivity of this type of assay, we can look at wild-type bacteria rhodopsin, and highlighted in black are the 3 states associated that I just showed you the structural states for. And then we can make a point mutant, or we can make a different point mutant. In a single record of a single molecule, you can see changes in dynamics of these proteins. Now, what we wanted to do is get energetics, and there are a number of ways in which we can do this. Perhaps our favorite this day is using a non-equilibrium assay to determine equilibrium energetics. What we do here is unfold and refold, and then analyze all the unfolding dynamics. So there are lots of transitions here. So it's a very information-rich type of assay. And then we use an analysis developed by Olga Dutko, because it's a coupled system, to determine the transition rate as a function of force. And one can measure the unfolding rate as a function of force and the refolding rate as a function of force. And you can, for any given transition, you can determine the F1/2. This is the force at which you would equally populate the 2 states that you're looking at. Conceptually, what you can do then is calculate the work that you do in going between these 2 states, but you have to use some correction factors to get the actually true underlying change and free energy. And so now we can do this for wild type, and we can do this for point mutants. And the reason to do that is we want to look at the energetic contribution from a particular side chain, or the so-called delta G. And that's what really biochemists are going to be interested in. So when we make that mutation for shown here, our AFM measurement gives us a delta G that's very similar to a prior measurement. Now, this point mutation faces inward, so it probes a protein-protein interaction. Now, when we, so that's very good, but when we do a different point mutation that faces outward, we see that the delta G determined by our assay is very different from the canonical SDS assay. And this is important because there's been a lot of concerns with SDS assays because they're not generally done in lipid bilayers. There are questions about SDS interpreting SDS to 0 concentration, as well as lacking native protein interactions. And so we were wondering what might be happening here, and my postdoc realized that there's a squalene here that interacts with this particular residue. And if it's strong enough to interact with that residue, well, it certainly has an energetic contribution, and that's what we attribute to the difference between what we measure delta G in native lipid bilayers and what one measures in SDS. So we think we've established a platform for quantifying the energetics of membrane proteins in their native lipid bilayers. So these cantilevers are not just good for looking at membrane proteins, they can be used for looking at globular proteins as well. So shown here is a collaboration with Marcelo Soso that was spearheaded. And so what we see here is a type 3 secretion system. And so the effector protein translates through the type 3 secretion system, but the tunnel here is only 2 nanometers wide, so it has to unfold as it goes through into the host cell. Now these effector proteins can rearrange the cytoskeleton and as well as change the immune response. And so an important factoid is that when we look at GFP or DHFR or ubiquitin, people have shown, not us, that when you take these proteins and you attach them to the effector proteins, they inhibit secretion. And so the paradigm for these types of proteins is that effector proteins are thermodynamically unstable. So when Marc-Andre looked at these two proteins, SOPy2 shown here in green because it's all alpha helical, or SBTP shown here, alpha helical and a beta sheet component, and computed the delta G, or the change in free energy upon unfolding, using canonical ensemble assays, he got a change in free energy of 6 to 7 kcals per mole. We can compare that to the free energies of unfolding GFP, DHFR, or ubiquitin, proteins that stall the type 3 secretion system. And what you can see is that they're very similar. So if you compare that to a library of other proteins of different structural classes, you can see that the effector proteins that we've studied here, SOPy2 and SBTP, have very typical thermodynamic energies. So in fact, effector proteins are not characterized by low thermodynamic stability. So we were therefore interested in mechanically unfolding these. So we adopted some technology developed by the Galb Lab and other people, and merged that with our modified AFM cantilevers to mechanically unfold these proteins. So you can see that the unfolding distance...
You have 3 minutes left.
Yep, thank you. You can see that the unfolding distances are rather large, which just to highlight that these are rather large proteins. We can apply this to both effector proteins that we studied and do the canonical analysis where we look at force unfolding as a function of loading rate. And the first thing that you notice that these unfolding forces are quite low. Particularly for SBTP, which has a beta sheet component, the distance to the transition state is rather large. And so we can compare effector proteins here with 3 other proteins which have been studied by single molecule force spectroscopy. And what you see is that effector proteins have rather large distance to the transition state and a low unfolding force. You can look at a number of different proteins that have been cataloged by Hoffman et al. And see that, in fact, there's this sort of relationship between F unfold and the distance to the transition state, sort of independent of the exact protein that you're looking at. And in fact, you can look at this sort of analysis with a Bell-Evans type of model. But the real take home here is that there's a new paradigm, which is it's really the large mechanical compliance of these effector proteins that facilitates a secretion. And so we're working on following up on this type of stuff. And in the last few slides, what I'd like to do is just highlight this RNA hairpin assay that I showed you before, that typically this RNA hairpin would be pretty hard to see by AFM. But when we look at the unfolding and refolding of just a short 12 base pair hairpin, we can easily see it unfold and refold. And by using site-specific attachment, we can drive this many times. And then we can, for a single molecule, we can actually measure the unfolding and refolding approximately 1,100 times, shown here, in 15 minutes. Now, this isn't 15, this is 1,100 unfolding events of the same individual molecule. And so finally, we can stop altogether and do equilibrium assays. The dynamics are fast, but you can actually resolve them. We can compute the probability distribution and use things like the inverse Boltzmann analysis to compute free energies. But what Michael Woodside has taught us is that we need to deconvolve the instrument. And when you do that, you get the classic asymmetric free energy landscape, but now for the first time derived in an AFM. So what I've shown you is we've used modified AFM cantilevers to do a variety of different types of single molecule force spectroscopy, but hopefully there are others here that are interested in adopting this technology and applying it to a diverse array of systems. And with that, I'd like to summarize. We've developed microsecond resolution cantilevers on a commercial AFM. We've uncovered hidden intermediates. You can apply this system to derive energy landscapes from equilibrium and non-equilibrium data. And we've determined energetics of membrane proteins. And you can apply these metrological advances to diverse systems. And so I'd like to thank my group members who've worked on this, our collaborators, and you for your attention.